Hi, welcome. Welcome, welcome, welcome. Hi, how are you doing? How are we feeling? Ready for paper three? I thought I would just jump on and give a bit of support because I had a bit of time tonight. So is there any specific topic areas you want to cover? Please do ask away. Excuse me. Hi, Tony. How are you doing? You're in year 12, but you hope the year 13s are doing really good. Ed, I have to say, the one thing I would say to you is make sure you're nailing your year 12 content now, because when you get to this point next year, love that, Tony, you're still alive. Good. Keep going, keep going. The key thing is making sure that you have AS content in the bag so that when you come to year 13, you're just enhancing that knowledge. How did I find paper one? It's an interesting one. It was a lot of enzymes. Hi, welcome guys. Lots of enzymes questions, which if you didn't know, that's what they were asking. So I went through and I did a big analysis of it again this morning. So obviously I've sat it as if I was a student because I want to see how I would do. And I always do that. Um, but as I was going through and looking at the topic areas that have come up, and I was writing my little notes to myself, um, yeah, it was a lot of enzymes, but hidden in the context of respiration and photosynthesis. I was expecting a lot on the photosynthesis side of things. Sorry, one second. I'm just trying to grab my little notebook. I was making notes this morning. Chromatography question paper. The question was really interesting actually because the chromatography is actually module two. So the TLC is module two. Because as we were going back over it this morning, I was like, where does it link? Which part of it does it link to? So the TLC was module two, but the questions relating to the darkness of it, that was practical skills. That was module one. Paper two killed you off. It's so hard. It's so hard when you're going through and you're trying to figure out what it has and hasn't done. So yeah, what did I put down? Um, yeah, it was literally, it didn't feel like a lot of modules that were covered, but there was in the Putnam Home because I only wrote about gorillans and the steps to start gorilla. I don't know, to be honest, I couldn't tell you. I've got to wait and see, and I can't remember. I mean, I sat the paper as soon as I saw it in the morning before I went live that afternoon. So I can't actually remember the question specifically. Um, And I don't know. This is the thing you have to realize the way that the mark schemes are, as I keep saying, the mark schemes, although they're sitting there ready and waiting, they're not fully finalized yet. So would that get you a full mark? I don't know. And that's the thing, you won't remember. You'll remember bits of the questions, you remember bits of your answers, but it doesn't necessarily mean that you have written exactly that in the wording that you've given. It's really down to the way it's written and what you've written. So I can't tell you, yes, you'll definitely get the mark point, or no, you won't, because I don't know. I'd have to read it in context. Oh, I'm sorry it was tragic for you. It's an interesting one with the neurons because the neurons kind of came up with regards to the depolarization, repolarization with the ECG question. So, in that sense, there was some parts about neurons, but it wasn't in the same way as what you feel that if they were asking you about the content of neuronal communication, like you guys want it to be that you memorize the information and then you're regurgitating it onto the paper. But actually, what happened was they gave you a lot of the information that you would memorize in a sense, and it is all application based. Everything that you're doing, there are a couple of 1-2 markers which are AO1. So there was one question that was literally stating about carbon dioxide, where would you find it? So that was an AO1 question. That was a 2 marker, really simple, complete recall, nothing more than that. Hold on, what? Sahi, were you actually given the wrong exam paper? So hold on, you were given a different exam board's paper, or you feel like you were given a different exam board's paper? Like, because it was so bad, it feels like it was a bad one. Yes, I'm not. I will cover that question. Give me 1 second. Let me just see. There's not been a question on drawing a graph. Yes, I feel like it's going to come up potentially on a histogram. I won't be able to see it. Miss Lauren, I got given a different maths paper for Edexcel maths paper too. What? Did they realize at the beginning? Yeah, I feel like if what I'm hearing right from students with regards to your paper too, I feel like that first six marker, because I haven't actually seen the paper, all I've got is what students have told me. But if it's anything like, um, no, are you kidding? Sahib, you were given the wrong paper. Hold on, there's a backup paper on accidents running theory. That's crazy. That will have to be noted and sent to the exam board. Also, Selena, if you had that fire alarm goes off, that actually has to be noted and sent to the exam board so they're aware that that happened. It doesn't necessarily mean that you will get any consideration, but what it does mean is that you'll be taken and should have been isolated and no conversations be had. We've had that a number of times. It's really annoying. Yes, you still sit the paper. You have to be taken to a secluded area where you can't speak to anyone, and the invigilators have to ensure that you haven't actually spoken to anyone. Your time stops, and then it restarts again when you're back in the paper. Wow, that's crazy, Zahib. I hope you managed to they managed to figure that out and that doesn't disadvantage you. But I would definitely be speaking to the college because I wouldn't be happy if that was my school or my children. I would not be happy about that. So I would definitely go back. That's interesting because I've never heard about the contingency papers. It wouldn't surprise me, but I've never heard of them because so we've never had to use them. But then if there are contingency papers, the only people who are aware of them are the exams officer within the school because they're the only people who get to see and lock down the papers before the actual exam. So anyone outside of that shouldn't be seeing or know about these papers. So that's an interesting one. Not that I've ever heard of that, but it doesn't mean it's not necessarily going to happen. Um, you don't know where histogram is. Oh, that's not good. If you go to yeah, basically measure the frequency. If you check out my live that I did 2 days ago, P, you'll see I actually talked about histograms there, and I give a little sneak peek of my booklet on it. I feel like I've done horribly in both the other papers. I hope you haven't, but you don't know for sure that you haven't actually done well. The way you remember things will be different to what you've actually written. So I wouldn't worry about it because you can't change it. Just move on and try and focus on the next one. I know. Well, this is the thing. I wasn't sure whether to bother going live next. I know a lot of you do chemistry as well, and you have your chemistry coming up on Monday. But then I was like, well, I could sit and watch a bit of trashy TV, and I was like, oh, but I feel bad that I could also offer my help. So I thought I'd jump on here and do a bit of support. Um, It's hard. If you don't know enough to write, then it's what do you do? And the thing is, as I said in the time before, just write anything you know, and you may pick up some marks. Don't stress about how much you've written. I am very blunt and to the point with my writing sometimes, and therefore I won't write a lot, and then someone will write an essay's worth, and we could still get the same marks. So I wouldn't stress out about the amount that you've written, but be very careful with what you write. That's the key thing I would say. So bad after hearing other, don't care about Candy, don't care about anyone else's answers, and I mean that in the nicest possible way. Every one of you will write things in very different ways. Oh, hold on a second. I need to plug you in. Definitely helps if you've got your power cable there. Amna, sorry, I forgot. You did ask me to go through a question on the paper, and I'm just talking away here. Um, Amna, what was the question you asked for again? Let me just check. 2023, question one, paper three. Right, let's have a look. Let's have a look and actually do some biology whilst we have the time, because I could just talk for Great Britain and talk about anything and everything. So let's have a look at some content. Should you redo all content? I wouldn't advise doing all content. What I would advise is picking the content you struggle with the most and work your way through that one would be my advice, because that's going to be the difference between you. Oh, interesting. Right. So it's question one, right? Oh, no, I don't want that. Give it away. That is not what I want. So here we go. Let's excuse me. Was it this one? You should be coming up in a second, I'm hoping. Just double check. You said 2023, question one, paper three, please. Yep, 1AI. Good. Okay, so this one's an interesting one. It actually, I would say, isn't a nice question at all, because this question is completely throwing you off, because what it's saying to you is you have got a trachea. Your trachea is being shown in your like micrograph, and you're being told that you're looking at the tissues. And it wants specifically to note what the tissue labeled A and B are. So this one, excuse me, is a really interesting question, because what it's looking at is, do you actually know what is made up in your trachea, in your exchange systems? So the fact is asking about that is basically checking, do you really know all the different structures? And if you don't know them, hold on, no, close that. Then what is it that you're actually looking at? Well, you should know that you have got cartilage that's going to keep the trachea open, and you should know that the cartilage is a C shape. And the reason it's the C shape is because you don't want it to be closing the airways. The airways need to remain open, and because they need to remain open, that allows for the air to flow in and out. So that ventilates the lungs. So that's the first thing you should know, is that there has to be some cartilage there, and there has to be that cartilage is going to be keeping it open. So when we look at this one then, and we're looking at, well, what is A? If you know, and if you think about this, your trachea is a hollow tube which is kept open by these cartilage rings, and these cartilage rings are going to basically do that. So therefore, what we're actually seeing when we look at C is already showing us our crumbs. Let's try and write that again. Ciliated epithelial cells. So the fact it's showing us epithelial cells, that would therefore tell me that what I'm looking at is B, first off, is my cartilage, because it's the bit which is quite a large shape and it looks like it's going to go all the way around. So that's where I would say that this is the cartilage. And therefore, I know not only do I have cartilage, I also have smooth muscle. So that would therefore tell me that this would be my smooth muscle if I were looking at that question there. It's mammals. No, it's not just in humans. Remember, when you're looking at the ventilation system, you're looking at the mammalian ventilation system. So yes, insects don't have it, but we're looking at cat, and you're already told in the question that cats are mammals. So that was their little hint to you in that question. Uh, does that help? I'm not going to lie. I do feel like there is histology coming. So if I'm wrong, please correct me. But I don't feel like any histology came up in paper. Well, it definitely didn't come up in paper one. So paper two, I would say that because paper two hasn't actually um, got any I haven't seen it. If it has got any histology, then that would indicate that potentially, yeah, I didn't think so. So I feel like it's coming up. And if it is coming up, then that would make me assume that that could be the one to do with muscles. Maybe. Again, I don't know. I mean, I have I don't want to call them predictions because I don't feel like they're predictions. I feel like they're more guesses. So with regards to a question that potentially is going to come up, I've put out my topic areas I believe are going to come up, but I could be getting this completely wrong. I don't know. So I'm not saying only study that. Make sure you are covering as much as you can. But the key thing is to know how to link these together. That's what's going to get you the marking point. If you understand your topic area, you will know how to link it together. Okay, so let's look at this one then. So what have they told us in this question? Oh, come on, wait 1 second. Right, here we go. So we're looking at the nervous system of a cat and how it coordinates a response. Right, so I know it's my nervous system. I've been told I've got a sensory receptor at the back of the throat is going to detect the stimulus. Well, they've basically given me all my keywords here to start with. They've told me it therefore goes through a sensory neuron. We've now got a spinal trigeminal nucleus in the medulla oblongata. Then we've got the relay neuron. Then we've got the medulla oblongata. Then we've got the motor neuron, and then we've got the muscle contracting. So this is obviously the response. The muscular contraction is therefore my effector, is my muscle. My muscle is going to carry out the response. So I've got a stimulus that has been detected. That is pressure on the throat. So therefore, if we're thinking about this to do with pressure, just which other way could they go? What we'd be saying is this is a mechanoreceptor because it's to deal with pressure. And we can see it's going sensory neuron, relay neuron, motor neuron, and then our effector that's going to carry out the response. So it's asking us to explain two conclusions that can be drawn. So it's going to the medulla oblongata. It's not going up. If we think about the brain, it's not getting up to the cerebrum. So remember your parts of the brain that you should know. So you've got your cerebrum, you've got your cerebellum, you've got your hypothalamus, and you have your pituitary gland, and then you have your medulla oblongata. So you should know that your medulla oblongata is to do with involuntary responses. And so the fact that we've been told that it's the medulla oblongata here I mean, it's giving me extra words that are freaking me out a little bit, and I don't really like to say the words that I'm like, oh, because I get myself all tongue-tied. But what I am noticing is I've got some hints in there. I just needed to look for them. So the fact that I am not getting up to my cerebrum, that is therefore indicating that I am using involuntary responses. There is no conscious thought going on. Therefore, it has to be involuntary. There is no somatic nervous system being linked here. So, this is one thing that I can talk about. The other thing is, I've basically gone one neuron, two neurons, three neurons. So, I know that there is a synapse here. I know there's a synapse here. So, I'm basically having very few synapses. So as I've said before, it's not like it's going sensory, really, 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 all the way back down. It's basically going sensory to the medulla with a relay to the motor back down to the effector. So therefore, the reflex is going to be very fast because I'm only using three neurons here, because that's what I've been told, sensory relay, motor, and therefore I've got very few synapses involved. So it's a very fast one. Yeah, you're right. The paper one had the grana and the stroma with regards to a picture in it, but it didn't really have any histology. It had electron micrographs, but it didn't have the histology that I would be expecting them to throw in there. Um, Yeah, does that make sense? Perfect. So this is actually a really good paper in the fact that, so this was 2023. So they were looking at the animal response side of things and looking at the reflex arc in that question. So animal response came up, but not much in paper so far. It was more plant responses, whereas if you look at 2024's paper, 24's paper had more on the animal response to do with how you coordinated and were able to look at the concentrations of the likes of carbon dioxide and how that therefore meant you'd get a faster breathing rate, faster heart rate. So you can see that they're picking little bits out of the topic areas in each of the years. And so therefore that's where they're going to bring this in. Now, I did do a little bit this morning. And I was like, ooh, and I hadn't got around to actually sharing this. So the question is, where did I store it? Where did I put it? Where did I put it? Where did I put it? Did I put it in here? Oh, I don't know. I don't know where I put it. Give me 1 second. Oh, there it is. So I'm going to show you guys. This is going to make no sense to you whatsoever, but hopefully because you're here and I'm explaining it to you, it will make sense. So someone asked in one of my videos, because obviously I've put out a few videos today to try and help you guys on what I think may come up. And someone said, well, does that mean they haven't asked a question, or does that mean that they have asked the question? What does that actually mean? So I'm going to show you a little bit of my analysis. Now, this is just the long questions, okay? So if we were looking at paper 2023, and I've taken out the multiple choice, I've got that separate, but I haven't included it here. So I went through every single question on the 2023. I've done it for every single paper, but I've gone through every single question, A, AI, AII, AB, and so forth. And so therefore I've made my notes of what I found and what those questions were based on me doing my quick analysis. So we can quite clearly see heart came up, paper one, cardiac output, and so forth. So this is what was each of the questions, the long questions in paper one in 2023. So then if you go to paper two in 2023, what you'll notice as you look through these is there was no parts in here that are coming up in this paper here. So basically the aspects in paper one, So remember, you've got module 1, module 2, module 3, and module 5. And then paper 2, you've got module 1, 2, 4, and 6. Now, I will bring this up to you. So 2022, paper 2, 2022, paper 2, had a part in it which was fill in the gaps on Hox genes. So I just want to show you something here, which is 2023's paper 2, nitrogen cycle, fill in the gaps. So one of the things as you're going through and you're looking at the questions. Now, these questions could be a 1 marker, they could have been a 2 marker, they could have been depending on what the question was. And I have all that in my analysis. But you can see eval-ev basically means here's some evidence, now evaluate it. Oh, improve the conclusion, what that conclusion was. So this was 2023, paper 2. And then 2023, paper 3. So what I did is as I went through, they had, and here again, I'm going to show you paper 3, 2023, homeobox and role of genes in brain. So what you can see is the questions can come up again and again and again, but how they ask the question changes. So 1 year, what they might do is they might ask it as a fill in the blank, and then they change it into a question where you have to apply your knowledge. So it's about understanding what they do and what they don't do. So when you see a fill in the blank one, and if it's on biological molecules or proteins, that's because remember, they have to give you questions which are 3 different levels. There's a low, there's a medium, there's a high. When you're being assessed, now you don't see any of that, you don't know any of that. And actually, teachers don't really know that unless they are really going through and analysing these questions. So you won't see it in the mark schemes, but there are questions of different levels. And therefore, what they're doing is they're giving you some where you will be able to get the marks, and they'll give you others where you're really applying it. So therefore, that was paper 2023, my analysis of it. So you can see 2023 had a TLC. And it had a calculate your RF value. So if that's the case, then why have you got another TLC? Well, because it's a practical skill, that they're assessing your practical skills on that. Now, one of the things that you'll notice 2023 paper 3 was a genetic modification data DV, dependent variable. So it was literally just a explain genetic modification on the data you've been provided. Talk about patents and so forth. So that may make people go, oh, they asked about genetic engineering, so they're not going to ask about genetic engineering again. But remember, it's about how they ask those questions. It's about how they interlink it that's going to make the difference. So when we looked at 2024's paper, when I went through, there was so much on the likes of urine analysis, all to do with the excretion topic area, homeostatic mechanisms, plant hormones, but they were tropisms. Hence why I said to you before paper 1, I've got a feeling germination is coming up because they haven't asked about germination in a while. So it's things like that that you can use to guide you, but they're also going to be asking you things like RQ. Well, you were asked about an RQ. So the thing is about photosynthesis and photosynthesis and respiration is so huge, it has to come through. If they ask you about the nitrogen cycle, they ask you about the carbon cycle, they have to be talking about photosynthesis and respiration. If they are talking about fermentation within the biotechnology, they have to talk about respiration, but it's about how they ask those questions. Never want to hear Gibran again. 2024's paper 2, so there was all these information that you had bioinformatics, there was evolution genetics in there, and yet it came through again. However, one thing they did note when it came to ecosystems was so much on succession. So therefore, do I think succession's coming up again? There may be a small 1-2 marker, but I don't think it's going to be huge. And if you look at paper 3 on last year's one, you will note that there was a genetic modification biotechnology question, but it was a 1 marker. So there's so many ways they could go with that. So it's really fascinating when you do this analysis and you go through and you're looking at where they've asked the questions. It is very much they can go any way they want. Okay, so just be aware on that one that they can go any which way they want. And so therefore it's important that you don't get hung up on these topic areas on their own, that you understand that they can go any which way they want to go. Because they may start talking about root hair cells, but actually they're assessing you on your understanding of transport across a member. I still feel that's coming through as well. Well, this is the thing. Paper 3 will be what paper 3 will be. All you can do is make sure you take the time to figure out what exactly it is they're asking you. So if I just randomly pick, I'm going to just pick it, a paper 3. I don't know which one. It will be the first one that I click on. So it may be one you've seen, it may be one you haven't seen. So let's just go on this one. I'm going to randomly pick one. I'm going to go to a question. No, don't want that one. Of course, I would pick. Okay. It'd be the ones that. No, we're good. They just rolled over. Um, just rolled over. I've picked a rubbish one, so I've picked one that's basically all the stuff that you guys have already covered. Typical. 2018 is the things I don't think are going to come up. Let me see. Let me see 2019. These are the next one on the list. But actually, I feel like eventually. Let's just look at this one. Oh, actually, I love this question. Do I love this question? I do, but I don't think it's coming up, so I shouldn't do that one. This is typical. I'm finding all the ones that are the stuff I don't want to because they've already covered all that. Nope, nope, nope. I'm not going to ask that. I'm not going to ask that. They've asked that already. Oh, maybe this one actually. Right. There we go. Let's have a look at this one. Oh, excuse me. Right. So if we look at this question then, one of the things to note, I really do feel muscles is coming up, and if I'm completely wrong on this one, it will be really interesting to look at what I've predicted in that. But I feel like it's only been asked once in 1 year. There's just so much more to come that I feel like, yeah, I feel like it is coming, but I could be wrong. Uh, I do feel like if the translocation comes up, I feel like it will be one of those ones that will be application based. It won't be straightforward. It won't be just a memorize this. It'll be linking it to other things. Like I feel like amino acids, nitrates, because the loading that goes into the phloem is not just sucrose. So I feel like they may throw you a curveball and tell you that amino acids are moved in the same way as the sucrose is moved, and therefore they're expecting you to describe how that process is, but linked to amino acids. And I do feel like because they haven't done, if I'm right, correct me if I'm wrong, did they do anything on plant diseases in the paper? Because I feel like that could potentially have a nice link in as well. Right, let's have a look at this question. Yeah, no human diseases. So human diseases came up last year, which would therefore make me feel like they're not going to ask you something simple like give us the T cells and the different types of T cells, because that was last year's paper. So therefore, I feel like it's going to be something more obscure or looking at plant. They do seem to be going down the plant side of things this time. Yeah, the antibiotic resistance is a good way of bringing in your natural selection evolution along with communicable diseases, because the antibiotics are obviously used to fight against bacterial cells and the bacterial pathogens. Um, so yeah, so that's the way I would think it's going to go. Right, let's have a look at this question then. So you're being told about an organism. Oh, no, I did not mean to do that. Let's try that again. Um, two organisms. So we've been told about two organisms, and we've been told they reproduce sexually, but under particular conditions, they can reproduce asexually. Well, how interesting is that? So I've just randomly picked a question, and yet it sounds similar to some bacteria. Bring in how translocation to and how into. So off my head. You're talking about that. And I watched a program last night called Beth, which was all about that, which was really fascinating about. And that's where a lot of students struggle when it comes to paper three. It's that they don't understand that when they are giving you this information, they want you to take your time. They want you to really decipher what it is that it is being asked. So that's something that I would therefore be looking at and saying, right, what are they telling us? We've got these diploid cells that are going to detach from the body of the sponge. The cells reattach to a surface and grow into new. Well, I know they're diploid ones. They're doing asexual reproduction, so therefore I know that that is going to be through the process of mitosis. Um, Asexual reproduction, in this case, they're using meiosis. Meiosis is about forming gametes. So therefore, I need to help myself to understand what's happening here. I'm producing gametes. I've got two haploid cells produced by meiosis are going to fuse to form diploid. Well, we're going to therefore end up with variation. The diploid cells develop into a new shark. And there we go. Clones, both animals produce clones. This is a I can agree because of this. I would disagree because of this. And so, therefore, when you see an evaluate question like that, a lot of the time it's about saying, right, what have they told me? They've given me hints here. They've said diploid cells reattach and grow into new ones. Therefore, I know that that is mitosis, and therefore that is genetically identical cells. So if you are given, and this is again why I think it's really fascinating. So I've literally just picked a question. I picked a question from what year did I say that one was? A random one from 2022. And what have they done? They've taken a very similar question, just reworded it and given you a different take on it, but it's the same thing. If I'm writing what I've been told from students about what came up. So this is what you've got to understand. They will never ask you the same questions again. And this is where students get hung up on memorizing mark schemes and thinking that that's going to help them. It's not going to help you. What's going to help you is you being able to apply your knowledge. So I'm going to bring in one little part and I want to show you how and all the different ways that I feel like they could go with a topic area. So if I were wanting to think about where might they go, if I'm right in what I've said, they haven't mentioned anything about the genetic engineering. They haven't mentioned anything about fermentation. And therefore, I can quite easily go, right, okay, so they haven't told me anything about that. So where could they go? What might they ask me about? Okay, so a lot of you, give me 1 second, I'm going to show you where I want to go with this. A lot of you will have potentially memorized this. Oh, hold on, let me do it that way. So genetic engineering, what do? They take a plasmid from a bacterium. They can isolate the gene mRNA, so they can either use restriction endonucleases to cut the gene and isolate it, or they're going to have the mRNA, they're going to do reverse transcriptase. Reverse transcriptase is going to make complementary DNA. Great. We have got our plasmid, our vector, which is going to be cut open with these restriction endonucleases. And what we're going to end up doing is we're going to end up sticking these together. So some students will memorize this content and be like, yes, I've memorized it. I know it. I could answer a question on this. And they throw a curveball at you and they say, okay, how do you know whether you have managed to fuse the plasmid and the complementary DNA or the gene together using ligase? So instead of testing your knowledge on the fact that it's a vector, the fact that it's a plasmid, the fact that it's ligase, the fact that you've isolated the gene, how are you going to actually be able to know that there has been a successful fusion, that you have got a recombinated plasmid? Because remember, you're going to take this recombinant plasmid and you are going to insert it into a host cell. You're going to insert it by electroporation. Again, I've memorized this. I'm ready and raring to go in this exam, that if they ask me a question, I know exactly how to answer this question. But that is maybe not where they're going to go with this. What they may do instead is they may say, okay, how could you do this? So if I were to take gel electrophoresis, not fluorescent tags yet. I'm not putting that recombinant plasmid in yet. I want to check whether it has been successful before I put it into my bacterial cells. And this is actually something I did with my students. So we used to get in Amgen Bruce project. So if you've ever heard about the Amgen Bruce project, it was connected to Cambridge University. It's an American company that basically loans schools equipment so that they could do genetic engineering of, obviously, the genetic engineering part of it was very basic and it was nothing that would be dangerous. But what we did is we took the plasmid, we took the DNA. So they had already given us the fragments, and we were then sticking them into the sample tubes and seeing if it was being successful. Well, to check if it was being successful, we actually had to do electrophoresis. So if I have got gel electrophoresis, because they haven't asked anything on this either, if I know my DNA is negatively charged, I know it's negatively charged. I therefore need to make sure that I put my wells where I'm going to put my DNA close to my negative electrode. You'd be surprised how many students did it the wrong way around and got no results. Um, Again, that could be a question they ask. Why did the students not get this? How could you be sure? How could they see? And so that's one of the things for you to note. These are real situations that people do and they make mistakes. So you need to be thinking about how to improve, making sure the wells where you're putting your DNA that is negatively charged, is close to the negative electrode. So going back to my original question, how could we be sure that we had recombinated the plasmid? And that is by knowing that our plasmid that we have extracted will have a size. That size, I'm just going to make this up, let's say is 1,000 base pairs. And let's just say the gene that we've cut out, excuse me, is 70 base pairs. So if I were to have three, let's start with three different samples, A, B, and C. If I were to put in a restriction enzyme with my plasmid and nothing else, I would expect to be able to get a fragment that I'd be able to visualize using gel electrophoresis, PCR before, and then gel electrophoresis, which was 1,000 base pairs. And that would indicate my plasmid. Now, my 70 base pairs, I'm going to put that isolated gene in B. And therefore, because it's a lot smaller, I would expect it to move a lot higher on my profile and therefore be up at the top. So now what I can see is I've got 1,000 base pairs, which is quite close to the well because it's quite large, and 70 base pairs, which is further up. Well, if my plasmid has been recombinated, that therefore means that this now is 1,070 base pairs because they have come back together. So if they've come back together, that will therefore mean that you'll be able to see that this would actually not go as far because it is bigger. And so because that's got 1,070 base pairs, that's therefore going to be a bigger sized plasmid because it's recombinant, because it's got an extra bit of DNA inside it. It's going to be bigger. It's not going to move through as far. And therefore, I'd be able to identify that as being successful. If, however, I have another sample, which is D, and in this one, I end up with two bands, that would prove that this is not successful, that these have recombined, and therefore I've got my plasmid and my extracted DNA separated and not together. So this is a way in which we could prove recombinant DNA has occurred. Now, it was interesting because there was a question on the success rate of a recombinant plasmid being taken into the cell. I haven't seen a question about, not on OCR anyway at the moment, on how you could actually visualize that, but that's a way in which you would be able to visualize it. By doing gel electrophoresis, you can therefore bring together genetic engineering with PCR and gel electrophoresis so that you can be sure that you have successfully recombinated that plasmid. So then what they could do is they could then go on and talk about the fact that what they'll do is after they have successfully grown the bacterial colony on an antibiotic medium, they can then move it to the fermenter. So this is where the visualization would be. By putting it on the antibiotic medium, you're therefore ensuring that they have actually got the correct one. So they know because it's got an antibiotic resistant gene. So this is fascinating that they had a big question on antibiotic resistance in your paper too, because obviously placing the bacteria so that they grow, population growth curve, because that hasn't come up yet. Correct me, guys, if I am wrong on that one, but you haven't been asked about the lag, the log, the stationary, and the death phase. So if you haven't been asked about the growth, population growth curve yet, this could link with genetic engineering, with fermenters, and with how you would put it into a continuous fermenter. Thank you, Beatrice. A continuous fermenter, and therefore, where would you be wanting it to be? Yeah, that one log calculation that I saw, was that paper one? I don't know if I remember right. That was quite an interesting one actually. And so therefore, this is where they can interlink it, like I've been saying, because that actually covers so many different parts of the topic area. Do you understand that a plasmid is DNA, circular DNA, module 2? Do you understand that you need to remove the DNA either through using enzymes or mRNA doing reverse transcriptase, module 2 plus module 6? So it's how they could assess lots of different topic areas in one big swoop of a question, all under the process of doing fermentation. Yes, absolutely, Anna. Let me have a look. Hold on, I think I've still got it open. What question did you say? Question 3, AII. Excuse me. 3A. This is a good one. That's a good one as well. No, which one did you actually say? Yeah. Where have you gone? Where have you gone? Where have you gone? AII. Um, Let me just bring it up. Here's all my notes of when I did that with a student the other day there. Right, okay, so genetic modification and selective breeding. What are they telling us? Genetic modification. Oh, no, move that. Did not mean to do that. Let's try that again. So genetic modification, selective breeding can be used to improve the characteristics of crop plants such as maize. So high light intensity, photosystem 2 will absorb excess light energy. A process called non-photochemical quenching converts the excess light to heat energy. Okay, just going to make a little note. Heat energy, kinetic energy, fire rate. MPQ can continue when light intensity is no longer in excess. This makes photosynthesis inefficient. Scientists genetically modified, excuse me, genetically modified crop plants to limit. Right, so we're going to limit MPQ because it continues. It's no longer in excess. Okay. So instead of us getting light, we're going to end up with heat that will be lost potentially to the environment. Okay, what else we got? Scientists exposed unmodified plants and GM plants to a period of high light intensity, followed by lower light intensity. Okay. Scientists then compared the rate of NPQ, the rate of carbon dioxide fixation. Right. So rate of carbon dioxide fixation, we know that this is the Calvin cycle. Because we know this is the Calvin cycle, we therefore know that this is the light independent reaction. The light independent reaction relies on products from the light dependent reaction. So therefore, we need light to be absorbed so that we can make ATP and so that we can make reduced NADP. Okay, so that's, again, I'm just making little notes to myself to try and figure out what I already know. Right, rate of NPQ. So let me just go back to this. So NPQ converts excess light to heat energy. Okay, so excess light is going to be converted to heat energy. Heat energy is going to be lost to the environment. We're going to have kinetic energy, potentially a higher rate, but not necessarily. However, if light intensity is no longer in excess, it will continue. If it's continuing that, and there's no longer light in excess, then that's going to affect my light dependent reaction, because instead of me having light, it's been converted to heat and therefore decreases the rate. Okay, so the light dependent reaction, not getting light because it's been converted into heat energy. Right, let's look at the actual results. So unmodified plant, genetically modified plant, time after. So time after decrease in light intensity. So it's 0, then 150. So we can see unmodified and genetically modified rate of NPQ was 1, but after 150 seconds after, we have, as you can see with my little arrows, a decrease and then a double decrease. And interestingly, what you're seeing, I've actually snapshotted straight from the paper when I sat this, as if I was one of the students. And so this is little hints that I do when I'm going through and analysing this, not as I'm doing it right now, but as I'm doing it as a pretend student. I will literally look at this and I will start drawing arrows that tell me if it's down. If I put one arrow, that's my little way of saying, right, that's dropped, but it's not dropped as much as the other one, hence why there's 2 arrows. However, I'd actually say there's not very much in it between those two. I would need to do a statistical test if I really want to test it. But anyway, that's beside the point. And then again, you can see unmodified to modified compared to afterwards. We have a decrease more in the unmodified one with a rate of CO2 fixation rather than with the other one. So explain the effect of genetic modification on the rate of CO2 fixation after 150. No, so what you're saying is, with regards to the MPQ increase after 150 seconds, because light intensity is no longer in excess, you're talking about the fact that in the unmodified one, it tells you that the non-photochemical quenching converts excess light to heat energy. So it's looking at the rate of it. So if it's no longer, it will continue even when light intensity is no longer in excess. So the scientists exposed unmodified plants and genetically modified plants to a period of high light intensity, followed by lower light intensity. So what you're seeing here is after the high light intensity, the rate of MPQ was 1. The rate of MPQ dropped down with both unmodified and genetically modified. However, it dropped down more with the genetically modified plant. So therefore, if it dropped down more with the genetically modified plant, you could, in a case, argue the fact that that is going to mean there's more photosynthesis occurring in the genetically modified plant. Therefore, there would be a higher yield because there's less MPQ than in the unmodified plant. So the MPQ rate is the rate at which it's going to convert excess light to heat energy. So it's higher when there's a high light intensity, but it will still work afterwards. Does that make sense? I can't, I'm afraid. Um, no, do not write arrows. Be honest, do not write arrows in the actual answer. I'm doing that on the table of results, but I would then write that in the actual answer. I'd write this one has decreased, but this one has decreased more. That would be how I would answer that question. Oh, no, hold on. I didn't like that. 1 second. There we go. So yeah, don't do arrows up and arrows down. Yes, Candy, let me just finish this one and then I'll go over zero dilution because I haven't done zero dilution and they do seem to be more and more, and it links back to the question about fermenters, etc. But let's just explain the effect of genetic and the rate of carbon fixation after the 150 seconds. So does that make sense on how to answer that question now? So the effects of genetics on the rate of CO2 fixation. So I talked about the rate of MPQ, but now we're looking at the rate of fixation. It's decreasing, but not decreasing as much as the unmodified plant. And the reason being would be that there is going to be more light that's going to be used in that light dependent reaction, therefore more products, therefore more carbon fixation. So it's that interlinking part of it that's going to make the answer or the way to get the marks in it. You can tell my sessions are never pre-planned in these ones because I just talk absolute drivel. This is definitely not what my actual sessions are like. Okay, so I got asked a question about zero dilution. Let me see if I can find a quick one on zero dilution. Excuse me. I'll be glad when you guys have finished your exam just so I can have a rest from my voice. It's that whole thing of I want to help as much as I can, but my voice is definitely taking a beating. Right, okay, let me think this one through because they did that in 20. Yeah. Yeah, so the fact that they asked you to do that way in 2024 makes me think that if they are going to ask you about it, um, oh, actually, let's do this one. Okay. I want to, I want to do this one because this one again links to, and I don't know why I'm going down this route, but I'm going to stick with it and I'm going to just deal with the backlash if I'm wrong in what I'm saying. But this was a paper three. Okay, so this was paper three, 2023, still in the same paper. Actually, if I've got, let me have a look, because that paper's often was. Thank you on this question. So, I've talked about the fact that you are that they've covered so much, or they had so much on it, but basically it's related to everything. So if we were to look at this question, let me just anaerobically, and I shouldn't investigated how respiratory. Um, in the yeast. Now, they're talking about for different populations of yeast provided with these different respiratory substrates. So this question's really looking at your understanding of respiration in the fact that you have got different biological molecules, and those different biological molecules, how they are affecting the respiration rate for the yeast. So my question would be, well, what if you were to put these yeast and ferment them, put them into a continuous fermenter and give them these different nutrients? Would that make a difference on the population growth curve? So this is where you've got to understand that when they are doing these types of questions, they're never going to ask it exactly the same way, but they will reuse different previous questions and just ask it in a different way. So they're talking about the total volume of carbon dioxide that's produced. So they're talking about growing these in the lab and checking how much they're actually producing that volume of the gas. So how did they collect that volume of gas? Did they use a gas syringe, for example? We don't know any of this information. I'm just asking the question. How did they collect that? So it could be, instead of them wanting you to talk about the biological molecules and the fact that they need to hydrolyze them down and they provide different monomers and how the yeast are able to utilize those monomers, because they kind of talked about that a little bit in paper one. What they might do is they might link it to how did they collect this data? What did they do before they moved them into the fermenter, etc. So then you've got this question where The student planned to compare the rate of anaerobic respiration. And again, correct me if I'm wrong, guys. I don't feel like they did anything on anaerobic respiration in paper one, where they could bring it into um, paper three, in the fact that if you had yeast cells that are anaerobically respiring, if they're anaerobically respiring, they're therefore going to be producing ethanol. Well, ethanol is really useful if we were going to be brewing, for example, to make alcohol. So how are we going to actually know which substrate to give them? Let's say, what is it that we want as our product? Whereabouts in their population growth curve do we want to be removing that? And how are we going to ensure it doesn't affect other cells? So it is important when you're doing this that you think about those linking parts. Okay, so this one then went on to say, let's try and do this, that you have, the student produces a 0.01 mole per decimeter cubed glucose solution for each of these population, and his method that they use produces 0.01 mole decimeter cube. Make two 10-fold dilutions from a 1. Just move that out there. Just move that out of the way. So straight away, you've been told that you have a stock solution. They produce a 0.01 mole per decimeter cubed glucose solution for each yeast population. And this is a method that they use to produce it. So this is what they've produced, and they are going to make a 2 times 10-fold. So therefore, what they're doing is they have 1.0 mole per decimeter cubed. They want to do a 2 times 10-fold. So therefore, they would take 1 centimeter cubed of the stock solution, and they would add in 9 centimeters cubed of distilled water. So if they're going to take 1 centimeter cubed and 9 centimeters cubed, they're going to have a total of 10 centimeters cubed. And that volume is going to be what they're looking at. So in the question, does it say millimole? It says mole. Mole per decimeter cubed. It doesn't say millimole, it says mole. So we've got a 1.0 mole per decimeter cubed. We're going to do a 10-fold, 2 times that 10-fold. So if I were to take 1 centimeter cubed of my 1 mole and add in 9 centimeters cubed of distilled water, I've now got 1/10 in my solution. So then I would do that again in each dilution using a measuring cylinder to measure the volume of water and a dropping pipette to transfer the glucose solutions. So they're basically asking you, do you know how to do a 10-fold dilution? With a 10-fold dilution, 1 plus 9. Each time mixing it to ensure that you are getting what you require, which is that 10-fold. So if I go on this one, ooh, excuse me, what I'm just doing is I am going right. What's my concentration? What's my volume? So if I've got 1 centimeter cubed and I'm going to have 1 centimeter, sorry, oh, I've just written 1 centimeter cubed. You can tell it's getting late. 1 mole per decimeter cubed. And if I'm going to take 1 centimeter cubed, I am going to divide by 10 because in total I've got 10 centimeters cubed in there. And so therefore, we are getting a 0.1. If I do the same again, because I'm going to do two tenfolds, I take 1 centimeter, let's try that again, 1 centimeter cubed down here. I'm going to add in my 9 centimeters cubed to get 10. I'm therefore dividing this by 10 again, so I get 0.01. So therefore, when you are looking at that type of question, if they're telling you bits of information, they're telling you that it's a twofold. They're expecting you that it's a 2 times tenfold even, that you're doing 1 centimeter cubed and adding in 9 to get to the concentration. And that concentration that we were wanting was 0.1 moles per decimeter cubed. So therefore, that's why I was doing a 2 times 10-fold in the question. But what I wanted to show you is this last part. Again, fill in the gaps. They love a fill in the gaps. So if they're doing a fill in the gaps, that's them doing the easy one, which means another time they're going to potentially ask it in a much harder way. So to avoid contamination, place the yeast in glucose solution that are produced using culture each species of yeast in different flasks. Ensure anaerobic conditions by using a flask. So you can see this one's quite easy in the sense that they are filling in the gaps. Where do I think they could go? Describe an investigation that you could do, that you would be able to culture these yeasts for moving into a fermenter, because that's basically what you're doing, right? So it's important that you note that whenever you see a past paper question which has been very much a fill in the blanks, that means another year what they're going to do is they're going to give you a question which is going to ask you to not fill in the blanks, to describe that process. Now, again, that doesn't mean it's definitely going to be the case. However, I would advise you to take the time to go through these types of questions and just look and see if you could answer that question. And what would you say? So do you understand that it's distilled water? Do you understand that they've told you it's anaerobic? So you need to ensure that there's no oxygen. So you're going to have to seal it. What are you going to seal it with? How are you going to ensure that there's no oxygen getting in? What do you need to do? What would you do to measure that rate of carbon dioxide? How would you measure it? What kind of equipment would you use? So that would be my advice and where I could see potentially them going. Guys, my phone went dark on me, which meant it is after. Yep, it's 10:05. I'm going to obviously finish so I can go to bed. And I have been going 79 minutes. Wow, I really do talk a lot. I hope that's been helpful. I will try and do another live when I get a chance to. So thank you for those of you who are obviously joining and sharing and liking. I do appreciate it. And yeah, just keep going. I know it's tough. I know who knows what they're going to ask? We can only guess, but it's about you being sure that you know your content to know how you can apply it. I definitely feel like whenever they do a fill in the blank, so go and check them out. Whenever they do a fill in the blank one, normally a year to 2 years later, they will turn that into not the same question, but one where you have to be the one telling them how you would plan something or telling them how you would do. So obviously the nitrogen cycle was a fill in the blank one. This fill in the blank one, so I can see where they may go with that question, but I might be wrong. Candy, I hope that helps with your zero dilution side of things. I don't feel like they're going to give you a question which, like they did before, where it was 22 colonies were growing and then working your way back. I don't feel like they'll do that this year because they did it last year. But again, I might be wrong on that. The plan came up last year. What do you mean the plan came up last year? Hold on, let me double check. When you say the plan came up last year, what did they ask last year? Let me look at my analysis. What am I on? Paper 3. So they did TEM, prokaryotes, estimating, Zero dilution improvement came up last year. Estimating population came up. Sampling, conservation, meristem, transpiration. Transpiration practical came up. Excuse me. Diabetes. See, now I'm like, was it in paper 2? Yeah, pedometer. Yes, yes, yes. So pedometer did come up. So it was a plan on how to investigate that, but that was on pedometer. Do I feel like pedometer's coming up this year? Absolutely not. I don't feel like they're going to ask you to plan a pedometer. What I'm saying to you is I feel like they're going to ask you to plan an investigation like this, which would actually be either using a form of culturing and making sure it's anaerobic, or it could be using a respirometer. So it's not going to be a pedometer, but it'll definitely go. There's definitely going to be one in there, which is plan, because that's module 1, along with assessing your content, but it's about the application. So this is why what I'm saying to you is knowing and practicing the past papers are great, but what if they flipped that question on its head and said, right, instead of you filling in the blanks or you analyzing this data, how would you plan it? How would you actually do a zero dilution so that you could count the number of colonies, for example? So do you understand that you need to do enough zero dilutions down, but not too many, that you have a good number of cells? So if you were to do, let's say, 7 zero dilutions, so the tenfolds 7 times, and in the final, let me actually show you what I mean by this, because that again is one of the things that I do feel like could, might not, but could. Let's say we were going to do zero dilutions and we were going to plate out each of the different ones. See, this is, I was about to go to bed, but I will finish this last thing. So if I were to do yeast solutions and I was culturing those yeast ones, and I was going to do zero dilutions, and each time I take 0.01 into my plate, 0.01. So each time I'm doing a zero dilution, I can't even do my arrows at the moment. This one, I can't even tell my colonies. This one, I can't really tell. I've got maybe a colony here. This one, however, I've got specific colonies that I can count. And here I've only got 4 colonies. Which zero dilution should I take, for example? Which one would be the best one to ensure that I could estimate the right population that I started off with? Because remember, if I am doing continuous fermentation in a fermenter, in a bioreactor, I want to ensure that I have it at the exponential log phase. If I'm doing batch, I want it to be at that stationary phase. I don't want it to get too large that it therefore gets to the point where I am going to end up with a death phase. So which one would I actually want to use? And the correct answer would be this one, because this one will give me a more accurate estimate compared to this one. My estimate would be way out if I tried to work my way back from that one. So that is another way where they could look at serial dilutions and working back on estimating how much was in. And again, because I'm linking it back to biotechnology, that's what I would have said. So again, I hope that's helpful, guys. I'm gonna go. Have a good evening. I can't believe it's Saturday night. Well done. And keep going. All I can say to you is keep going. You have 3 more days. I know that sounds crazy, only 3 more days, and less if you've got other exams still going. If you've got your chemistry on Monday, good luck for that one. But there's 3 more days. Oh, do you know what, H? If you really wish you could give me something back, you can write a review on my Trustpilot. That would be amazing because obviously I'm helping you guys as much as I can. And the reason I can do this is because of my current clients that I work with who actually pay for my time. And so therefore I need to, obviously when you guys disappear, I need the whole bunch of students to come. And I'm so surprised how many people still don't know who I am or about my resources that are out there. So that's what I would say. If you really want to give me something back, write some reviews for me, guys. So that the current year 12s know who I am, and know to find my resources, where to find them to help them. But have a good evening, and I will see you when I get back on here. I'll try and come on tomorrow night. I can't guarantee it, but I will try, and I'll be on. I'll do a bit on Monday after I've got my clients. I've finished with my clients. I'll jump on Monday, and obviously on Tuesday when I've got not got my clients as well, I'll jump on. So guys, have a good evening and I will see you.